Good morning, Adrian. I do want to just say I'm proud of you guys for maintaining good attendance so far this year. This has been a strong class. You all have been showing up and doing what you need to do. So that deserves recognition and praise and proud of you. Although I'd like to see some more cameras on. It's kind of weird talking to Mr. Krabs and 42 and a picture of Adrian's little brother, I assume. Thank you, Kiki. There he is. What's going on, Joseph? There's Gabby. Gabby, you've got to give me some background on the paintings. Is that all you? Did you do all those? Impressive. Are you selling any?
Yeah.
Okay. Well, we may need to talk. All right, about 3 minutes left to go ahead and do that warm up. Shouldn't be a long one. I'm glad to see some cameras on. Makes me feel good and welcomed and not alone.
Mr.
Was I on mute that entire time?
Yeah.
Wow. Okay. Thank you. Um, what I said is that today is going to be a review day of Unit 2. And today is the 5th day of our unit. So this was a really quick and short unit. We only have 4 days of new material. And today is going to be a review day. And then the 6th day, tomorrow, will be test day. So we really want to make sure that we have all of the opportunities possible to discuss everything we've talked about over the last 4 class periods. And so our goals for the day: biologists will be able to solidify their knowledge of the structure and function of cells. So I say solidify because we're not learning anything new. This is stuff that we've talked about. And then biologists will be able to clarify any and all misconceptions they may have about how cells complete the Stirringer processes. So as we move forward in biology, as we go from one unit to the next, we have to keep in mind that this is a cumulative course. So concepts are building on one another. What we discussed in Unit 0 when we first introduced Stirringer is still going to be relevant in Unit 2, it will still be relevant in Unit 5, and then it'll still be relevant in Unit 10. So we still need to be able to remind ourselves of what Stirringer stands for and how it's relevant in this specific unit about cells. And then the essential question that we're hoping to answer by the end of the lesson: what are the features of different types of cells that make life as we know it possible? All right, so I'll give you 30 seconds to make sure you have your notes out, to make sure you're writing down the title of the presentation. Today, I want to make sure that the energy is high. I want to make sure that you all have every opportunity to ask questions, taking yourselves off of mute to answer questions. And even if you're unsure about your answer, even if you think it could be wrong, I still want to hear from you because that will still benefit us to know what is one person confused about. Oftentimes, what one person is confused about is more than likely confusing to everybody else. And so that brave person who shares out an answer and then that answer is wrong is actually doing everybody a service, because now we know, all right, Mr. Rudd, you need to clarify this for everybody. So keep that in mind. Okay. So the first thing that we will talk about today, the first standard that we will talk about today is 1.1.1. And this standard is all about the organelles that we've discussed. And specifically, this standard says, summarize the structure and function of organelles in eukaryotic cells, including the nucleus, plasma membrane, cell wall, mitochondria, vacuoles, chloroplasts, and ribosomes, and ways that these organelles interact with each other to perform the function of the cell. So this is literally the language that the state of North Carolina gives to all of the schools, all of the high schools across the state, and says that students, in order to demonstrate mastery, need to be able to do these things. All right, so it's important that you all have an opportunity to see this language. And if you can check it off and say, yes, I can summarize the structure and function of organelles. Yes, I can summarize the way that organelles interact with each other. Then you know that you're in a good place to master the content, to pass the class, and to pass the EOC in January. All right, so I like to show you all that language to be as upfront as possible. But I know you all can do this. I know you have the knowledge, and I know you have the skills to do these things. So let's just jump into it. You don't need to write down this standard. All right, we've got 2 cells here, 2 different types of cells. They are both eukaryotic cells. Does anybody know which one is a plant cell and which one is an animal cell?
The green one is the plant cell.
The green is the plant cell. Good. All right. Now, I'm going to have some arrows here. Hopefully you all I don't want to be disrespectful if anyone is colorblind, but the colors will hopefully be some hints to you, and they're still arrows, so you'll see what they're pointing to. What is that organelle that the purple arrow is pointing to?
Nucleus.
That's the nucleus. Good. So we've got two nuclei. So both plant cells and animal cells have nuclei. What about those pinkish orangish things?
Mitochondria.
Excellent. Those are mitochondria. All right, so the way that we can identify mitochondria, well, let me take a step back. The way that we can identify nuclei is that they're typically pretty big and they have a dark spot in the middle. And that dark spot is specifically called a nucleolus, but we don't, as we saw on the last slide, we don't talk about that in this class. So you can see whenever you see a big thing in the middle of the cell and it has a dark spot in its middle and its core, then that's the nucleus. And then these mitochondria, they have these distinctive folds that look like a winding river in the middle of them. Okay, and that's important. Again, that's kind of above the level of this class, but that's an easy way to identify the mitochondria. They've got this winding river in the middle of them. What about these things, these light blue amorphous things?
Well.
Spell it. Spell it. Yeah. Okay. Vacuoles. Yes, you were right. Thanks, Gabby. Excellent. So these vacuoles, again, I say they are amorphous, meaning that they don't really have a defined shape. They look like these blobs. And one thing we should take note of is that the vacuole over here is way bigger than the vacuoles over here. We've got several vacuoles in the animal cell, but they're smaller. And only one vacuole in the plant, well, we do have several, but the main one is much bigger. Can anybody remind me of why that is? Why is the vacuole in a plant cell so large?
Because it needs to store in a lot of water.
Excellent. It needs to store a lot of water. Okay, this one's hard to see, but they're like these green dots that we see. What are those?
You said the spots?
Yes.
Ribosomes.
Excellent. So those ribosomes, they're a little bit difficult to identify because they're so small and they really can appear in many places throughout the cell. But oftentimes, as is the case in these pictures, you'll see that they are very close to the nucleus. And we're going to talk about that on the next slide, why that is. All right. Now the pink outer layer, what's that called?
Cell wall.
Not the cell wall.
Cell membrane.
The cell membrane, also known as the plasma membrane. Good. And I'm glad we said cell wall first because that's what this is, right? But one thing we should obviously take note of is that there is no there is no cell wall in an animal cell. Only plant cells have that cell wall. And then what's this last green thing all the way up here?
Chloroplast?
Yeah, that is a chloroplast. Good. And these last two organelles appear only in plant cells. Only plant cells have the cell wall and the chloroplast. All right. Does anyone need more time to see this? Simple head shake. Oh, he turned his camera off. Do you need more time, Kiki?
No.
Okay, good. Gabby, I can't tell if you need more time. Are you writing? Okay, you're good. All right. So again, we really want to focus, especially in today's review session, we want to focus on how these organelles relate to one another. So you all did a great job of identifying them, but can you tell me how they relate to one another and what their functions are? So the nucleus. What does the nucleus do?
Controls its organelle.
Good. The nucleus is the control center of the cell. So it's basically the boss. It tells basically everyone else what to do. Ribosomes, what do they do?
Make protein.
Excellent. Ribosomes produce the proteins. They synthesize the proteins. What about mitochondria?
Gives it energy.
Excellent. They provide energy. They are what we often call the powerhouse of the cell. They provide power. Chloroplasts, what is their responsibility?
Photosynthesis.
Excellent. They are responsible for photosynthesis. Again, chloroplasts appear only in plant cells. What about the vacuole? What is its job?
Stores water and nutrients.
Excellent, Kiki. Thank you. So it stores nutrients, waste, and water. The cell wall. What is the cell wall's job?
To give a plant structure.
It gives structure. Excellent. It's the rigid outer layer. It gives us structure and even protection. But I'm glad that you said it gives a plant structure because it only appears in plant cells. Thank you, Alicia. And then lastly, the cell membrane. What does it do?
Controls what comes in and out of the cell.
Excellent. It controls what can come in and what can go out. All right, so we know what they do individually, but how do they relate to one another? So what's the nucleus's relationship to the ribosomes? Any thoughts on that?
Does the nucleus tell the ribosome to produce protein?
It tells the ribosomes what proteins to make. And how does it do that? What are the instructions called?
DNA.
Excellent. Yes. So the nucleus provides DNA, which eventually leads to the production of proteins. So the nucleus gives the ribosomes their instructions and says, today you need to go make this. We can imagine there being a boss at a construction site who gives out work orders every day. And you have to go report to the boss before you start your work day so you can find out what exactly you're doing. What are you working on today? The ribosomes do the same thing, and that's why they're located close to the nucleus, because they need to be near the instructions. They need to get the DNA. All right. Sorry if it's getting loud in my background. What's the ribosome's relationship to the cell membrane? Any thoughts on that?
The proteins in ribosome makes goes past the cell membrane.
Excellent. Yeah. So some proteins need to leave the cell. Hormones, for example. Hormones are basically how our body's cells communicate to one another. So some hormones are produced in your brain, in the back of your brain, and they need to be communicated all the way down to your kidneys. And so cells in your brain will produce these hormones, and then they travel through your blood to cells in your kidneys to say, hey, we need to make some more adrenaline in this moment. Or we can have cells in the back of your brain that then get communicated to the front of your brain. So when you get tired, what happens to your eyes? When you're sleepy, what happens to your eyes?
Your eyes start to droop down.
Exactly, your eyes start to droop. And that's because there's a hormone that's produced in the back of your brain in an area, in a specific gland called melatonin. Maybe you've heard of melatonin before. You can actually buy melatonin over the counter, but it's naturally produced in your brain. And so once it's produced in this gland that's in the back of your brain, it can travel to the front of your brain and it starts telling your eyes, okay, it's time for us to block out the sunlight. It's time for us to start going to sleep. So your eyes start to droop a little bit. And so melatonin is actually the hormone that's responsible for maintaining what's called our circadian rhythm. It tells us when it's time to go to sleep and when it's time to wake up. All right. So yes, and long story short, Alicia was right. Some proteins need to be able to leave the cell. The cell membrane and the cell wall, how are they related? They both kind of share a responsibility. And someone said it earlier about what the cell wall does. I think it was you, Alicia. Okay, this one is tricky.
They both protect they both protect the cell.
Yeah, they both protect the cell and they provide the structure. They're both the outer layers. So protection and structure. Good job. Thank you, Amari. Okay, the vacuole. The vacuole is related to the cell wall. And this is something that we'll talk more about, later in the course. But there's an important relationship here. And hopefully you all have a chance to write this down. So if I'm moving too quickly, please let me know. Now, your pictures don't have to be all nice. We really just need to keep in mind how these organelles are related to each other. But the vacuole is related to the cell wall because it helps to provide structure. So the bigger the vacuole is, the more pressure I put on my cell wall. And if my vacuole is out of water, then it starts to shrink and I'm not putting so much pressure on my cell wall, and that's what creates drooping plants. So vacuoles need to store a lot of water so that they can place pressure on the cell wall. Any thoughts on how the vacuole relates to the chloroplast?
The vacuole provides water to help with photosynthesis.
Excellent. So this is again something we'll talk about in Unit 4, but water is needed for photosynthesis. You need water to make food. And specifically, the food that we're talking about is called glucose, which we talked about last unit. All right, so you can't make that food without water. So you need water from the vacuole to do that. Okay, how does the chloroplast relate to the mitochondria?
The glucose that's created from photosynthesis is then turned into energy.
Beautiful. So you need glucose, you need food to make energy, just like our bodies need food for energy. Plants operate in the same way. Cells operate in the same way. They need food to make energy. The chloroplasts make the food and they provide it to the mitochondria. And then last one, how is the mitochondria related to, or how are the mitochondria related to the ribosomes? If we need water to make glucose and we need glucose to make energy, then what's the next step of that?
We need energy to make proteins.
Beautiful, yeah. So the mitochondria provide the energy to make proteins. All right, so this is just a very basic representation of how these organelles are related to one another. And it's almost in a cycle of sorts. So I'll leave this up, take a screenshot of it, try to write it down. I'll leave it up for about 60 seconds. Okay. The next standard focuses on prokaryotic cells versus eukaryotic cells. The specific language says students need to be able to compare prokaryotic and eukaryotic cells in terms of their general structures and degree of complexity. Okay, so we talked about prokaryotic versus eukaryotic on day 2, which was Friday. I think today's day 5. Yeah. And we did this exact slide. So can we still do it? I don't think we got anything wrong the first time. Let's see if you all still remember. So plants, are they prokaryotic? Plant cells, are they prokaryotic or eukaryotic?
Eukaryotic.
They are indeed eukaryotic. Good. What about chromosomes? Do chromosomes exist in prokaryotic cells or eukaryotic cells?
It's the one that starts with a E, U something, E something.
Yes, ma'am. Eukaryotic, good. Okay. Which one, prokaryotic cells or eukaryotic cells, which one does not have a nucleus?
The one starts with a pro.
Good. Prokaryotic cells do not have nuclei. Bacteria. Are bacteria an example of prokaryotic cells or eukaryotic cells?
Prokaryotic. I don't know.
Good. That was perfect. Yeah, prokaryotic. Circular DNA. So DNA that exists in a plasmid, is that characteristic of prokaryotic or eukaryotic?
Pro.
Pro. Which one is older? Evolutionarily older.
Pro.
Pro, yeah. Pro came first. And which one has mitochondria?
The one that starts with an E. E.
Good, eukaryotic cells. Now we've got 3 images here that hopefully we can classify. This image, is it an example of prokaryotic or eukaryotic cells?
The one that starts with E.
Yes, good. That is a plant cell, and we can see that big nucleus right there in the middle. So that tells us that it has to be eukaryotic. If it has a nucleus, it's eukaryotic. So this middle image here, prokaryotic or eukaryotic? This one is also eukaryotic. We can see that there's a big nucleus right there in the middle. So when we see the nucleus, it's got to be eukaryotic. And then the last one, of course, is prokaryotic. So clear differences in structure between the two types of cells. And I want to just include a few more slides with some fun GIFs to explain it. So does it have a nucleus, prokaryotic, eukaryotic? Prokaryotic cells, no nucleus. Eukaryotic cells, nucleus. How's the genetic material organized? The prokaryotic genetic material looks like a bunch of spaghetti. It's very unorganized, it's twisted and tangled together. It has really no order, it's just random, and it's strewn throughout the cell. But the DNA inside of a eukaryotic cell is tightly wound in a very organized way, and that organized way is called a chromosome. Does it have membrane-bound organelles? So these are organelles that are surrounded by their own personal membrane. So that would be a mitochondrion, a chloroplast, or a vacuole. Prokaryotic cells do not have membrane-bound organelles, whereas eukaryotic cells do have membrane-bound organelles. And then lastly, I think this is the last one. Is it old and therefore unevolved? No, there's one more after this. Prokaryotic cells are very, very old, or as Kevin Gates likes to say, really, really. Eukaryotic cells, they're not old. They came later. They developed much longer after the prokaryotic cells. And prokaryotic cells are smaller, whereas eukaryotic cells are much bigger. And this is important because the bigger the cell is, the more complicated it is. And the last standard 1.2.3. So we only had these 3 standards in this unit. The last standard focuses on cellular adaptations. And the specific language says students should be able to explain how specific cell adaptations help cells survive in particular environments. And we wanted to focus on unicellular organisms. All right, so let's see if we can fill this out. So we talked about really 5 different types of cellular adaptations. The first one we talked about was an eye spot. What does the eye spot do? How does it help the organism to survive?
Can you repeat that?
Yes. So the first cellular feature or adaptation that we talked about was called an eye spot. How does the eye spot help the organism to survive?
It adapt behavior, moving towards or away from light.
Good. So they allow for light detection. They you can move towards or away from light, and that's what phototaxis means. So that eye spot is really just a small collection of proteins. It's not the eye that you and I have, or even the eye that insects have. It's very, very basic. All it does is allow the organism to know that area has light, this area does not. And depending on their preference, they can go over there or they can stay over here. Okay, so here we know that whatever cellular feature goes here, it allows for food ingestion. Does anybody know what feature that is? If you don't know how to say it, you can just spell it out.
The pseudopod?
It is. It's the pseudopod. Good. So those pseudopods are temporary projections of the cellular membrane. And they just kind of grow out in search of food. What do the cilia do? How do they help? How do cilia help the organisms to survive?
Is cilia the one with a lot of arms?
Yeah. Mm-hmm.
So it helps it to move and like feel around in its environment.
Good. Excellent. Yeah. So they can sense their environment, like Gabby said, and it also allows for movement as well. So we can think of cilia kind of like whiskers, but if the entire organism was covered in whiskers. They allow the cell to know, okay, this area is a little bit too salty. This area has too much hydrogen, it's too acidic. This area has too much oxygen, it's too basic. So based on that, the cell can sense it, and it can make a decision about what it wants to do with that information. Okay, what cellular feature or cellular adaptation allows for cell movement? What's the adaptation that allows the cell to move?
It starts with an A.
It does. It is the flagellum. Good. So it looks like a long tail. Some organisms have more than one. Some just have a single one, and it propels them forward in a specific direction. And then lastly, we've got contractile vacuoles. What do they do? How do they help the organism survive?
Stores and pumps out water.
Good. Couldn't have said it better myself. All right, so these are the 5 adaptations that we really need to know, excuse me. And we need to know we can think of it not about what they do, but how they help the organism to survive. So we've got to think that these unicellular organisms, they don't have multiple types of cells and multiple types of tissues and multiple types of organs that can divide up the job. They're just one cell, so they need to do everything for themselves. So these adaptations allow just that. They're simple, and they allow the cell to do many of the same Stirninger processes that our very complicated bodies do. Okay. And that's a good segue into talking about Stirninger. So as we wrap up the lesson, I always want to get in the habit of revisiting the objective. So one of the objectives for the day was biologists will be able to clarify any and all misconceptions they may have about how cells complete the Stirninger processes. So the S in Stirninger, you hopefully remember, stands for synthesis. So my question for you, and you can type it in the chat, you can unmute yourself, whatever you prefer. Where and how do cells synthesize new things? Where and how do cells make new things? Just provide me with an example of how cells make new things. No thoughts or people typing into the chat? No. I know we know this because we discussed it earlier. Okay, I'll repeat the question one more time in hopes that maybe you all just haven't heard me. Where and how do cells make new things? So all I'm looking for is an example of how cells make new things.
From other
Okay, so they can take things in from other cells and kind of repurpose them. But if I'm thinking about something that's brand new that the cell has to make, can we think of an example of that?
Cells making hormones.
Okay, so cells make hormones, which are a protein. So where do proteins get made?
Ribosomes.
In the ribosomes. Good. So that's all I'm looking for. That's an example of how cells synthesize. They, within their ribosomes, are capable of making new proteins. So that's a good example. We can also think about mitochondria, which make energy in the form of something called ATP, which we'll talk about in our fourth unit. So that's an example of them making new things. In plant cells have chloroplast, and they're able to make glucose using sunlight and water and carbon dioxide. Okay, so we just want to think about some examples of each of these. The T in Sterninger stands for transport. So how do organelles move within cells? And then how do some cells move towards and away from one another? Can we think of any examples of that? How do organelles move within cells?
Is there the flagella?
The flagella. Okay, so some cells have a flagella that allow them to move and swim in different parts. And then you were also trying to say cilia, too, C-I-L-I-A. Those cilia, like the whiskers, they allow the cell to move about as well. So that's how the cell itself can move. And then within the cell, we also have something called the cytoplasm, which we haven't talked about since day one of the unit. But the cytoplasm is a jelly-like fluid. So it's not quite water, but it's also not a solid. So that cytoplasm allows for ribosomes to move from just outside the nucleus all the way to the cell membrane. And they allow for vacuoles to move around and vesicles and all these other things that need to be able to access different parts of the cell. So the cytoplasm is extremely important for transport. Excretion. How do cells store waste, or where is waste stored?
In the vacuole?
Yes, good. That's all we need to say about that. Regulation. How do cells control their internal environment in response to changes in the external environment? What kind of stuff do they do to do that? I want to push your critical thinking skills here.
Do they communicate with other cells?
Okay, they can communicate with other cells. So they can sometimes get warnings about changes in the environment. Think about what one of the main purposes of a vacuole is in a plant cell. What's one of the main purposes of a vacuole in a plant cell? Seems we're getting a little tired. The lesson has gone on for a while. So plant cells, they have large vacuoles that store water. And that means that if the external environment, meaning the environment outside of the cell, if that environment becomes drier, then the cell still has a reserve of water inside of the vacuole. So it doesn't immediately die. It can't go without water for too long, but it can survive some period of time without water, thanks to the vacuole. So that's just one example of how they respond to their internal, or how they change their internal environment. Nutrition. So the question would be, how do some cells digest food? How do some cells make their own food? So we know that some cells have a pseudopod that allows them to digest food. We know that some cells also have, we haven't talked about this really, but specific enzymes that allow them to break down nutrients. But we also know that plant cells have to make their own food, and they do that with the chloroplast to make glucose. We'll talk about growth and development in Unit 5. We'll talk about reproduction in Unit 5. And then the last letter of Sternger is respiration. So this should be easy. How do cells make energy?
Mitochondria.
The mitochondria. Excellent. So everything that we've talked about today should allow you to answer this question. And I won't ask you all to share out, but just to kind of reflect by yourselves. What are the features of different types of cells that make life as we know it possible? So everything we've talked about today, between the organelles, excuse me, between the storage of genetic material in the nucleus or in a plasmid, between the storage of water, the creation of energy, the consumption of food, all of those things contribute to life as we know it being possible. So if you can really start to think about how those features are related and remember what those features are, then you are in a great place to master this unit. Okay, so we've got 20 minutes left, 21 minutes left in class, and there's not really much to do today. So all you really need to do is complete the exit ticket. And then what I would do with the rest of my time is complete all of the other unfinished assignments that there may be. And this is your opportunity to let me know, hey, Mr. Red, I can't access this, or this one's not working for me. And I can try to work around that, because tomorrow at 11:59, everything will be due. If you have done everything and you're caught up, that's excellent. I would encourage you to make a brain dump. So start to make sure that your notes are written in an organized way, and you can use that for tomorrow's exam. You can organize it in whatever way you think it would be most appropriate, but just try to get the most important information so that you can refer to it during tomorrow's exam. You don't have to search through all of your notes. That's what I mean by a brain dump. It's basically a cheat sheet, but it's not cheating because it's open notes. Okay, so I'll leave this up. I'll give you guys an opportunity to go back to Canvas and I'll be here on mute. Okay, folks, it is 11:35, so I will go ahead and let you all go. Please make sure you at least review your notes and take it a step further and study. Castro, I see you've joined us. Thank you. Study for our exam tomorrow. If you have any questions between now and then, I trust that you know how to contact me. And I hope you have a great rest of your day.
Bye.
Bye-bye.